Hello, my name is Olivia Colbert. I am a grad student at the University of Central Florida. Today I will be presenting on the role of CCR5 in HIV-1 transmission and treatment. I wanted to begin my presentation by reading the abstract of my paper. It states, "The paper reviews CCR5 and its importance and relevance to human immunodeficiency virus. As the global HIV positive population rose from 150,000 people in 1980 to nearly 10 million people in 1987, the need to understand HIV replication, transmission, and treatment became inexorable. HIV targets and weakens CD4 cells of a person's immune system by permitting the individual to become susceptible to secondary diseases and viruses. For HIV to penetrate immune cells, it adheres to and utilizes CD4, CXCR4, and CCR5 receptors. These receptors act as a passageway for HIV to transfer its genetic material into the host's CD4 cells. Through the progression of experimentation and research, a genetic mutation known as CCR5 delta 32 confers resistance against HIV by terminating CCR5 receptor expression. This discovery has led to HIV remission, as well as the development of therapies and vaccines targeting CCR5. This capstone/review aims to answer what key roles does CCR5 play in viral reproduction, transmission, and treatment, and why is it a critical determinant of HIV infection. Now, to begin, a little introduction on what the disease is that I'm speaking about. HIV is a virus that weakens a person's immune system by attacking CD4 cells, as I said a minute ago. CD4 cells are a type of white blood cell that protects the body from infection. CD4 cells stimulate B lymphocyte responses, which are responsible for producing antibodies, and T cells, which are responsible for cell immunity. When HIV attacks CD4 cells, cell immunity is compromised. A compromised immune system increases the risk of secondary infections that a healthy immune system would normally be able to fight off. When an individual acquires a secondary infection, also known as an opportunistic infection, they are diagnosed with AIDS, acquired immunodeficiency syndrome. This diagnosis can also occur when the CD4 cell count falls below 200 cells per cubic millimeter of blood. The normal range is from 500 to 1,200 cells per cubic millimeter. Since the pandemic began in the early 1980s, nearly 75 million people have been infected and 32 million have died of HIV or AIDS. 700,000 of the 32 million deaths have occurred here in the United States. Adult men make up 81% of the 38,000 new HIV diagnoses in 2017. In 2018, 46% of those diagnosed with HIV in the United States were Black or African American, 22% were Latin, 19% were persons of multiple races, and 13% were white. Now, to talk a little bit more about CCR5 and what it is, chemokines are a family of small cytokines, and cytokines are a large group of proteins secreted by specific cells of the immune system. They influence the immune system by regulating and mediating inflammation, by producing blood cells, platelets, and immunity. Chemokines regulate lymphoid organ development and cell migration as a part of normal tissue growth. They are also responsible for immune surveillance and immune cell recruitment. Chemokines are classified into one of four main categories. CC chemokines, for example, have two adjacent cysteines, whereas the CXC chemokine is separated by a single amino acid. I have an image at the top of the slide kind of illustrating that. The nomenclature, for example, the CCL5 gene is ligand 5 of the CC family of chemokines and CCR5 for its respective receptor. CCR5 is classified as a CC chemokine receptor type 5. So HIV participates in a 7-step process so that it is able to replicate and proliferate through the body. To begin, for HIV to bind, the viral glycoproteins known as GP120 and GP41 must interact and attach to the CD4 glycoprotein and chemokine receptor, CXCR4 and CCR5 respectively, on the surface of the CD4 cell. This fusion initiates a connection of the viral and cellular membranes, allowing the single-stranded viral RNA to enter the CD4 cell. Once the HIV RNA is released into the cytoplasm of the host cell, reverse transcriptase converts the genetic material from single-stranded viral RNA into double-stranded viral DNA. To do so, first, reverse transcriptase makes a DNA copy of the viral RNA genome. Reverse transcriptase then degrades the original RNA strand. A complementary DNA strand is then added, which results in the double-stranded DNA. This DNA is then moved to the nucleus of the CD4 cell. The viral integrase enzyme integrates the newly formed DNA into the host cell's DNA. Building blocks for future HIV are made when RNA is synthesized by RNA polymerase from the DNA. Protease, another enzyme, is then able to create functional proteins forming a virion. This new virus particle is then able to detach and locate another CD4 cell in the body to begin the process all over again. HIV transmission and ways to prevent transmission. So HIV is predominantly spread through bodily fluids during unprotected anal and vaginal sex, breast milk, and sometimes sharing injection drug equipment. Having protected sex and limiting the number of sexual partners are the best ways to reduce the risk of contracting or spreading HIV. Those at a greater risk of acquiring HIV should consider pre-exposure prophylaxis medication. This utilizes antiviral drugs as a strategy for HIV prevention. Post-exposure prophylaxis is an emergency antiretroviral therapy option to take within 72 hours of potentially being exposed to HIV, and although it is not 100% effective, if taken correctly, it may prevent HIV from replicating. Due to the absence of an HIV or AIDS vaccine, utilizing HIV treatment may prevent transmission. Two potential ways to prevent HIV transmission is through rectal topical application prior to anal sex and through the insertion of vaginal rings prior to vaginal sex. In a 2017 study, 5p12 Rantis showed to block the principal coreceptor CCR5. Another study showed that Rantis had significant stability after exposure to aggressive protease activity. The CCR5 inhibitor now is considered at the forefront of preventing heterosexual HIV transmission. A 2019 study showed that with a controlled release of the 5P12 rantis, it remained present in both vaginal fluid and vaginal tissue after a 28-day treatment. This could potentially become an effective way to administer HIV treatment as viral prevention. Beginning of HIV treatment. So in 1964, AZT, called azithromycin, was founded. AZT was eventually shelved after yielding ineffective results as a cancer therapy drug. It is also known as zidovudine. When the HIV epidemic began in the early 1980s, AZT was tested and was able to actually reduce viral replication without damaging any normal cells through blocking reverse transcriptase, the enzyme that converts HIV genetic material into double-stranded viral DNA. In clinical trials, AZT was also able to prolong survival and decrease incidence of opportunistic infections. AZT was approved by the Food and Drug Administration to treat HIV in 1987, soon after patients began experiencing symptoms like anemia, insomnia, and headaches. So because HIV replicates at such a rapid pace, it began to experience error and mutation known as drug resistance mutation. So when minor changes were made to the virus, the HIV variants became resistant to AZT. Currently, antiretroviral therapy is the most common form of HIV treatment. The goals of antiretroviral therapy, or ART, are to prolong survival, improve overall quality of life and immunity, reduce the risk of transmitting HIV to others, but the main goal is to decrease the HIV viral load to an undetectable level. Highly active antiretroviral therapy is a triple drug form of treatment that utilizes 3 different drugs to treat HIV. Now here I have inserted a chart that I made showing the different HIV drug classes, the different steps of the HIV life cycle that it interferes with, and the action that it takes. So for example, post-attachment inhibitors affect the binding process, so CD4 receptors are blocked from interacting with the virus. Fusion inhibitors obviously are, the fusion step is affected. It blocks HIV from entering the CD4 cell. Nucleoside reverse transcriptase inhibitors interact with the reverse transcriptase step. Reverse transcriptase is blocked from converting the HIV RNA into DNA. Integrase inhibitors, the integration step is hindered. HIV integrase is blocked from integrating. In my opinion, the most interesting part of the CCR5 gene and receptor is the gene mutation. The gene mutation was discovered in 1996. It is able to protect cells from being infected by HIV. It's able to do so by inhibiting the receptor expression on CD4 cell surfaces. Although the origin of the gene mutation is unknown, it is present in 10% of those of European descent and 20% of the Ashkenazi Jewish population. The primary difference between the gene and the gene mutation is the deletion of 32 base pairs of nucleotides 794 to 285. The mutant allele contains a total of 215 amino acids compared to the 352, as well as the inclusion of 7 novel amino acids. Thus far, the CCR5 delta 32 gene mutation is the only known HIV resistance mutation. It's also known as the only gene that has cured 2 HIV-positive individuals. In 1995, Timothy Ray Brown, also known as the Berlin patient, was diagnosed with HIV and began taking antiretroviral therapy a year later when it was discovered. Nearly a decade after his HIV diagnosis, he was diagnosed with acute myeloid leukemia. This is a type of blood and bone marrow cancer. It generates abnormal platelets or white or red blood cells. Having sufficient white blood cells is important to fight off viruses and bacteria, especially for someone that is positive in HIV. And having sufficient platelets and red blood cells is important for sufficient oxygen supply to tissues and to form blood clots to prevent excessive bleeding. Well, trying to rid him of his cancer, his doctor had recommended a stem cell transplant, and this is not uncommon for this type of cancer. The doctor's hope would be that after the transplant, Brown was rid of not only of his cancer, but of the HIV diagnosis. To do so, he needed to receive a stem cell transplant from an individual that is homozygous for the Delta 32 gene mutation. Once Brown had undergone a conditioning regimen, the transplant could commence, and ultimately it was successful. Not only was Brown cured of his cancer, but he was cured of HIV. In this slide here, I have incorporated an image from his doctor's research article. Lane 1 shows the CCR5 gene. Lane 2 shows CCR5 and the gene mutation. Lane 3 is just the gene mutation. Lane 4 is the patient prior to the stem cell transplant. And lane 5 is the patient day 61 post stem cell transplant. So as you can see, lane 3 and lane 5 are the same, meaning that he no longer had the gene, just simply the gene mutation. Unfortunately, in September of this year, it was announced that his cancer had relapsed, ultimately taking his life. But his vulnerability and forthcoming attitude prompted a huge change in the world of curing HIV, and in my opinion, his contribution will remain unmatched and always respected. Curing HIV for a second time. So the second person to be cured of HIV is Adam Castillejo. He is also known as the London patient. He was diagnosed with HIV in 2003. He was then diagnosed with Hodgkin's lymphoma in 2012. Similar to the last patient, Timothy Brown, he received a stem cell transplant from a CCR5 Delta 32 homozygous donor. The report of the London patient identified the central role of the CCR5 receptor, eliciting hope and anticipation for a cure, since he was also cured not only of his cancer, but of HIV. Now, once these two patients were cured of HIV alongside their cancer, many started to wonder why more HIV positive patients are not receiving this same type of treatment. So to begin, the stem cell transplants were done simply to cure them of their cancer, not to cure them of HIV. Curing HIV was simply an additional benefit. Only 50% of stem cell transplants are successful. The procedure alone is very aggressive. It poses many risks such as blood clotting, fluid retention, nausea, infection, jaundice, post-surgical pain. The outcome of the procedure is not as clear-cut as they want it to be. Rejection, known as graft versus host disease, is relatively common in bone marrow transplants. If this occurs, the new stem cells begin to attack the patient cells, possibly causing for a failed transplant. And these are just a few of the reasons why hoping to cure HIV with the stem cell transplant is not a reasonable choice for the majority of patients. Now, the story that brought my attention to the CCR5 mutation, basically creating my passion for the topic of HIV and the CCR5 gene, was the story of genetically modified HIV-resistant babies. In 2018, a biophysics researcher, He Jiankhu, announced that he had utilized CRISPR-Cas9 to genetically modify 7 sets of embryos. Each embryo came from a couple in which the father was HIV positive and the mother was HIV negative. 2 of the 7 embryos resulted in successful pregnancies and deliveries. Now, he justified his actions by stating that he was simply giving these children a lifetime immunity from the AIDS-causing virus. But this premature act was criticized for being ethically wrong, and the potential benefits are not worth the risk. But ultimately, this decision has modified the future of gene editing. A 2015 study showed that individuals with the CCR5 delta 32 gene mutation are at a higher risk of death after acquiring influenza. Another study that monitored brain injury following occlusion showed that the absence of CCR5 increased motor deficit and the size of infarct. After this story came to light, the United States put a ban so that the government would regulate the genetic modifications of embryos, sperms, and eggs. In the summer of 2019, the House of Representatives have restored this US ban on genetically edited babies. So the future of CCR5 research, what's next? So a few years ago, biologist Richard Pastel interviewed with Serious Science, speaking about his research that was to find the mechanisms that metastasize cancer, hoping to block it, essentially. The purpose of his primary study was to turn off what he called the cancer GPS. So doses of a CCR5 inhibitor identical to the doses administered to HIV-positive individuals were injected into mice with breast cancer. The HIV entry receptor blocking drug was sufficient to block the metastasis of the breast cancer. So then the second phase of their research was to determine if CCR5 is overexpressed on human cancer cells. In a certain type of breast cancer called luminal B, CCR5 was highly overexpressed. Once again, this led into administering inhibitors of CCR5 to determine if the breast cancer would metastasize. In the preclinical trials, the same doses used in HIV-positive individuals proved sufficient enough to dramatically decrease the number of cells that would spread to the lungs from the breast cancer. CCR5 is also important in inflammation. CCR5 directs leukocytes, a type of white blood cell, to sites of inflammation, and now studies are focusing on the relationship between CCR5 producing microglial cells and neuroinflammation. So to conclude my presentation and my capstone paper, CCR5 has transitioned from being a negligible receptor and gene into a pivotal finding for HIV. Researchers and scientists are now understanding how HIV enters a CD4 cell, ultimately leading to ways that can manage and hopefully eventually treat the virus. The gene mutation alone has opened doors for potentially curing HIV and the future of gene editing. The results and finding of my paper and of this presentation illustrate the role of CCR5 in different segments of HIV-1 and the importance of the receptor. So I wanted to just take a moment to thank you Dr. Cole and Dr. Clark for your assistance in this process. Dr. Cole, I don't know if you know how much your feedback has pushed me in this process. You have allowed me to see the potential that I have in writing such a intimidating paper that when I was told I had to do this, I honestly thought there was no possible way I could do so. But in your class, I was able to write about HIV a little bit, and in Dr. Clark's class, she taught a little bit more about HIV, and I already had a small passion for this topic, and you have pushed me to do more research and to understand more, to comprehend more, to be able to articulate more than I really ever thought that I could. So I wanted to thank you for your help. It genuinely means more than I could say. And Dr. Clark, thank you for being along this journey with all of the rescheduling and the constant emails and the uncertainty. Thank you for teaching me about this topic last spring, for helping me find a pathway for the topic that I wanted. Your help has really meant a lot to me also. So thank you to the both of you. Thank you to the University of Central Florida for giving me this crazy assignment that I never thought I'd be able to finish. I am now more confident to move on to the next parts of my life. So thank you. That is my presentation. I look forward to your questions, Dr. Cole and Dr. Clark, and I'll see you soon. Bye-bye.